All right, y'all. How's it going, nerds? So today our objective is nerds will be able to describe the structure and function. Notice that it is both. Here we go. Let's make sure we got our pen selected, our structure and our function. And it says of subcellular components and organelles. Subcellular just means anything that is smaller than the size of the cell. The component is something that is part of that cell, and then the organelles. Now, we're not going to focus as much on each of the individual organelles as we are going to focus on the membranes of organelles because A, they don't show up in every organelle, and B, they're really important in how these organelles function and why they actually are useful to the cell. So here we have our prokaryotic and eukaryotic cells, or examples of them, right? So here on the left, we have our prokaryotic. On the right, we have a plant cell, which is a type of eukaryotic cell. Plant cells, animal cells, fungi, all of those are going to be eukaryotic cells. Now, there's a couple of things that are similar. You're going to go through the lesson, but we see our ribosomes here. We see some cytoplasm. And then we also see a cell wall and a cell membrane here. The cell membrane sits directly inside that cell wall. Cell walls for structure. In the plant cell, we also see the same thing. Plant cells have cell walls here. That's this yellow stuff here. And that's for structure. And then right inside that, you're going to see a cell membrane. And then we'll also have some ribosomes here kind of pointed in these little dots there that you can see. Now, something that is different, though, is that prokaryotic cells do not have membrane-bound organelles. What that means is the organelles that are inside of here, right, none of them are going to have a membrane. Whereas eukaryotic cells on the other side can have plenty of membrane-bound organelles. We have chloroplast, mitochondria. I'm circling them as I go. This is the Golgi apparatus. This is the endoplasmic reticulum here on the outside, and then this is our nucleus. All of those have membranes, and all of them are able to do their jobs because they have these membranes. So chloroplasts are a great example of an organelle that has a membrane, and in fact, chloroplasts have double membranes, which is something they're going to share in common with mitochondria. Now, having all of these membranes allows us to do something called compartmentalization. Compartmentalization. And you can note that compartment is a major part of that word. So compartmentalization means that having these membranes, so here we have an outer membrane, that's the dark green one. Then we have kind of this lighter membrane that goes inside that, that's the inner membrane. And then even the thylakoids are basically made of a membrane, and they're kind of hollow in the middle. So that yellow lumen right here, that lumen is a compartment. And in that compartment, the chloroplast can actually store different things. In this case, they're really good at storing H+ ions in that space. And having a lot of H+ ions can actually be used to generate energy in the form of ATP. So having all of that energy in there, that H+ ions stored in there and kind of trapped, you know, it's a compartment. It's like a closet. They can put it in there and hold it there. And in this case, they can actually use it to produce some energy. Well, now mitochondria are similar. Again, you're going to see that we have our outer membrane here, that's the dark red. And then you have this inner membrane here, which is this kind of yellow color. Well, now you have different compartments, right? You have this space here that I'm highlighting, and it goes up in here too. That space can be filled, and the mitochondria is going to do the same thing. It's actually going to put H+ ions into that space, and once it fills it up, it can actually use it again to make energy in the form of ATP. But you'll notice that, you know, the inner membrane right here, I'll use a different color, the inner membrane in here also is a compartment, and you'll notice that mitochondria actually have their own DNA, and it's actually different than the DNA in your cells. They've got a few ribosomes. So having these spaces allows for these organelles to be more efficient, allows them to do different reactions, and allows them to hold things in different places rather than it all just like kind of running around in one big space. So that's compartmentalization. Big thing, right? Chloroplasts and mitochondria do that. The other thing they both do is something called, or they're increasing their surface area. So surface area is simply, let me get rid of all these lines now. Surface area is simply this idea that you have space right here, right? that is a surface, right? It's not just a line, it's actually a surface, right? Kind of like the inside of a container. And on that space, you now have a bunch of areas, right, where you can have chemical reactions occurring all along this line, just like a maze, right? Just like a container. Well, having all of these folds, and they're called cristae, but having these folds increases the surface area. Having those folds increases the surface area. Okay, that's fine, great. But why do they do that? Well, one of the advantages of having that increased surface area is now you can have more chemical reactions, because each of those areas is now a place where you could have enzymes that are doing chemical reactions. You could have ribosomes producing proteins. You could have proteins that are acting as channels. So the more surface area you can build in there, the more you can do whatever process you're trying to do. In other words, surface area, right, makes you more efficient. That increased surface area makes it more efficient. And chloroplasts have that too. So something that we're seeing in chloroplasts and mitochondria, and you'll see it in other organisms too, or other organelles too, like the Golgi apparatus and the endoplasmic reticulum, is this greater surface area through the use of these folds. And having those folds, right, means that you can now be more efficient as an organelle and as a cell, use less energy, take up less space, and you're still able to get what you need to get done. All right, and last but not least, having the folding of that membrane or having a bunch of compartments increases the surface area, and increasing the surface area means more efficiency. Remember, if you just had a straight line like that for a compartment, okay, fine, you could, do chemical reactions along the edge like that. But if you fold it like this, right, and you get that membrane really going, notice it doesn't take up that much more, well, it does because my drawing is bad, but it doesn't take up that much more space. But now you could have chemical reactions or transport or whatever happening all along here, which means in the same amount of space, you could have way more chemical reactions or way more transport, thus making you way more efficient. All right, so that's been our organelles video. Make sure that you had taken notes during this time. If you didn't, go back, pause it, put these in your notebook, keep on to these. These are our main key ideas right here. Have a good one.